Okay, this begins the second chapter in genetics, and we're going to begin to talk about chromosomes and cellular reproduction and the mechanics of cell division because it's important to understand how genetic material is passed on from generation to generation and also from cell to cell. What you're looking at here is fluorescence electron micrograph of a rat kangaroo kidney cell undergoing mitosis. And essentially, this is the process whereby genetic material is replicated and a copy of the complete set of instructions for running the cell end up in each new cell, which allows us the reasonable assumption that the new cells will look and act like the original, unless there's been an error in the reproduction of DNA, which we call a mutation. Okay, the green there are protein fibers that are called microtubules. Okay, and they are attached to the centrosomes of chromosomes, and this is a cell that is transitioning from anaphase to telophase. Okay. Now, when we talk about the the different organisms on the planet, living organisms, their their living structure, the smallest unit of living matter we know is the cell. But the cell comes in two different types of architecture, prokaryotic and eukaryotic cells. A prokaryote is a unicellular organism that has a simple cell structure, and these include bacteria and archaea. These are all single-celled organisms, okay? And they're quite primitive. And they're thought to have arisen on the planet long before the more, let's say, sophisticated cellular organization of a eukaryote, which is made up of a membrane-bound cell with a nuclear membrane, membranous organelles, and non-membranous organelles. Um, Eukaryotes are one of the three primary divisions of life, consisting of organisms whose cells have a complex structure that we've already detailed. Eukaryotes are both uni and multicellular, okay? Whereas prokaryotes are always unicellular. We can see the cellular architecture here, okay? Um, Prokaryotic and eukaryotic cells differ in structure. The nucleus in a prokaryotic cell is not really a defined region. It's simply where the genetic material is found, okay? But it is not bound by a cell membrane. In eukaryotic cells, there is a true nucleus with a nuclear membrane surrounding the genetic material. Cell diameters relatively small, between 1 to 10 micrometers for a prokaryote, and for a eukaryotic cell between 10 and 100 micrometers, almost an order of magnitude larger. DNA in a prokaryote is usually one circular DNA molecule that is not complex with histones in bacteria, but there are some histones in archaea, whereas in the eukaryote, they are complex with histones. The amount of DNA in a prokaryote is relatively small, although it is significant compared to the size of the cell. So it makes up a significant percentage of the cell, but the total amount of bacteria is, total amount of DNA is not extensive. Okay, whereas there's lots of genetic material in a eukaryote, and the reasonable assumption there is that the architecture being more complex in the eukaryotic cell, it takes more information to maintain and produce it. Okay, and all of this is coming from the DNA. Okay, amount of membrane-bound organelles in a prokaryote, there are none. In a eukaryote, examples of membrane-bound organelles include transport vesicles, peroxisomes, lysosomes, mitochondria, chloroplasts, rough and smooth endoplasmic reticulum, and of course the nucleus. Okay, just to name a few. There are also non-membranous organelles, such as centrioles and centrosomes and ribosomes. Okay, and there are the cytoskeletal elements, the intermediate and microfilaments and the microtubules. Okay, which are also not bound by membrane. So you can see the difference in the structure in these diagrams, and this chart sort of lays out um, the differences that we've been discussing here. Okay. Um, it's interesting to note that even though the prokaryotic and eukaryotic cellular architecture is dramatically different, that the two types of prokaryotes are not equivalently distantly related to the eukaryotic cells. It turns out that as a result of comparing the genetic sequence of archaea and typical bacteria to the DNA sequence of eukaryotes, we find out that the archaea are more closely related to the eukaryotes than the bacteria are. Okay, so one of the three primary divisions of life is called a prokaryote, consisting of a prokaryotic organism called a eubacteria. All right. In archaea, one of the three primary divisions of life, we have unicellular organisms with a prokaryotic cell structure, and then in the final division, the eukaryotes, okay, we have the membrane-bound nucleus and the more complex architecture. Okay, so that's our prokaryote, right? No membrane-bound organelles. The prokaryotic DNA isn't surrounded by a nucleus, and it consists of eubacteria and archaea. Now, that said, the term prokaryotic literally means primitive nucleus. Karyo means nucleus. Okay, so there is a region where the genetic material is found, but it's less clearly defined than in the eukaryote. These were thought to be the first living organisms on the planet. Okay, the photosynthetic prokaryotes were the leaders in the march of life, in that they were able to, as a result of photosynthesis, change the composition of the atmosphere, produce oxygen as a byproduct, and essentially set the stage for the evolution of the rest of the life on the planet. Okay, without the production of oxygen, the planet would look quite different than what it does today. Okay, so it was the early photosynthetic prokaryotes that are thought to have eventually evolved in some cases into chloroplasts, that provided the um, the food for many of the non-photosynthetic organisms that would evolve later, in the form of simple sugars and complex carbohydrates, and also began to change the composition of the atmosphere and the chemical composition of the what was at that time a global ocean. Okay, and again, this happened billions of years ago. Okay, a eukaryote is a different type of organism. Okay, it has a more complex cellular architecture. There can be both uni and multicellular eukaryotes. Examples of unicellular eukaryotes um, would be found in the kingdom Protista. Okay, um, you can see them if you take a sample of river water or pond water and look at it under a microscope. Okay, you can see a lot of these single-celled organisms moving around, some of which are photosynthetic, some of which are not. Okay, multicellular eukaryotic organisms, you see them every day. Okay, humans are multicellular eukaryotic organisms, bats, monkeys, fish, and so on. Okay, their genetic material is associated with proteins that are called histones in a membrane-bound nucleus. The histone's purpose is to compress the DNA into a smaller volume and thus protect the information from shear stress. But it's also thought that it serves a regulatory function in terms of controlling um, when units of information in the DNA called genes are turned on and off in the process of transcription. Okay, so you could think of these histones similar to the devices that are used to roll up garden hoses in your backyard for the wintertime so they don't crack when they're left out loose. But also sort of like entry and exit ramps off of the highway to tell you if you're a protein that's going to be involved in gene expression, where to get on and where to get off of the double helix. Okay? Viruses are very different. Okay, a virus isn't technically considered to be a living organism. All right, a virus is essentially either an active or an inactive particle, and its genetic material can be either double or single-stranded DNA or double or single-stranded RNA, depending on the type of virus. Okay, it's basically a protein coat, sometimes with lipid components, that surrounds genetic material at its core. And then sometimes inside the protein coat are specific enzymes that are unique to the ability of the virus to reproduce itself. Okay, and often those enzymes can serve as drug targets. Okay, it has a non-cellular structure. A lot of viruses they look like lunar landing modules or Christmas tree ornaments. Their basic design is to invade healthy cells and convert them into virus factories so that more virus particles can be emitted and other host cells can be infected. Okay? Because the virus is not a living entity, it can tolerate a greater degree of mutation than a living organism because it doesn't have to maintain genes involved in reproduction, okay, that living organisms utilize. Okay, it just has to have intact genes for the coat proteins and for the enzymes that are responsible for producing and packaging the genetic material, whatever it may be. Okay, so that's unique. Okay. Prokaryotic cell reproduction undergoes a process known as binary fission, okay, which is similar to a process in eukaryotes that we'll talk about in a moment called mitosis. Okay, when a prokaryotic cell reproduces, its circular chromosome replicates and the cell divides using the process of binary fission. Replication usually begins at an area called the origin of replication, which is a specific sequence of DNA in the circular chromosome. This process isn't terribly well understood. The origins of the two newly replicated chromosomes move away from each other toward opposite ends of the cell. In at least some prokaryotes, proteins bind near the origins and anchor the new chromosome to the plasma membrane at opposite ends of the cell. And then finally, a new cell wall will form between the two chromosomes, producing two cells, each with an identical copy of the chromosome unless there's been a mutation. Under optimum conditions, this can happen every 20 minutes, and at this rate, a single bacterial cell could produce 1 billion descendants in just 10 hours. Okay, so this origin of replication is a unique feature of the chromosome, where the enzymes that are going to produce the new DNA, the DNA polymerases, are going to bind. Okay, there will be enzymes that will unwind the DNA double helix. There will be enzymes that will create the new leading and lagging strands of the new DNA molecule. Okay, so let's take a look at binary fission.
Bacterial cells generally increase in numbers by the process of binary fission. The cell increases in length while doubling all of its parts. then forms a cross septum and divides to become 2 cells. 2 cells become 4. Those 4 become 8, and so on. This pattern of increase results in exponential growth of the population. The time required for the number of cells in the population to double is called the generation time. Depending on the medium, temperature, and other conditions, some bacteria can double in numbers every 30 minutes. Other bacteria have generation times of several hours or even days. During growth of the cell, streptococci and some other gram-positive cocci synthesize new cell walls in the septal region. Rod-shaped bacteria such as Escherichia coli, Salmonella, and Bacillus species synthesize new cell walls over the entire surface of the cell.
The entire sequence of events of cell elongation and binary fission, by which one bacterial cell becomes 2 cells, is referred to as the cell cycle. Because each daughter cell must receive at least one copy of the genetic material, DNA replication and cell division must be closely coordinated. Septum formation is triggered by attainment of a threshold length and mass, and the completion of DNA replication. A number of proteins, including the FTSZ protein, are essential for forming the cross septum at the center of the cell and not at its ends. These proteins are scattered throughout the cell between divisions. At the onset of septum formation, FTSZ protein forms a ring at the division site. The FTSZ protein ring is a contractile structure that uses the breakdown of GTP as an energy source. As the ring contracts, synthesis of cell membrane and wall follow until cell division is complete. The FTSZ protein and other cell division proteins then disperse throughout the cell. DNA replication occurs on replisomes, which consist of DNA polymerase, helicase, and gyrase not shown, and are probably attached to the cell membrane. When Escherichia coli is grown at 37 degrees centigrade with a generation time of 60 minutes, it takes 40 minutes for the DNA molecule to replicate. This is followed by a 20-minute period during which a septum is formed, resulting in cell division. No DNA replication occurs during this time. If the generation time is 50 minutes, a newborn cell contains DNA that has been replicating for 10 minutes during the end of the previous cell cycle. After 30 more minutes, DNA replication is complete. No DNA replication occurs during the next 10 minutes. A new round of DNA replication begins 10 minutes before the cell's complete cell division.
Now, in eukaryotic cell replication, we have a different sequence of events. Similar to prokaryotic cell reproduction, eukaryotic reproduction requires DNA replication, copying, separation, and division of the cytoplasm, but the presence of multiple DNA molecules requires a more complex mechanism to make sure that one copy of each DNA molecule ends up in each of the new cells. Eukaryotic chromosomes are separated from the cytoplasm by the nuclear envelope. The nucleus is highly organized with scaffolding that makes up the nuclear matrix. It's a network of protein fibers. The matrix maintains spatial relationship among the components of the nucleus and takes part in DNA reproduction, the expression of genes, and the modification of gene products before they leave the nucleus. So, essentially, this matrix stabilizes the information, but also plays a role in the expression of that information. So, any damage to matrix proteins can have an effect on gene expression or the maintenance of the information itself. Okay? Each eukaryotic species has a characteristic number of chromosomes per cell. Potatoes have 48, fruit flies have 8, humans have 46. There appears to be no special relationship between the complexity of the organism and the number of chromosomes per cell. However, there is a relationship between the complexity of the organism and the complexity of the information. Okay? which means that there's a lot of information in some organisms that doesn't necessarily contribute to the maintenance and the reproduction of that organism. Okay? In most eukaryotic cells, there's two sets of chromosomes. The presence of two sets is a consequence of sexual reproduction. Each set is inherited from the male and the female parent, and each chromosome in one set has a corresponding chromosome in the other. Together, the two chromosomes are going to form homologous pairs. In humans, as an example, you get 23 chromosomes from your mother, 23 from your father, making up a total of 46 chromosomes that form 23 homologous pairs. Okay? The two chromosomes of the homologous pair are alike in structure and size, and each carries genetic information for the same set of hereditary characteristics, generally speaking. But the sex chromosomes are an exception, and we'll talk about them later. As an example, if a gene on a particular chromosome is going to contribute to a trait such as hair color, another copy of that gene, known as an allele, at the same position on that chromosome's homologue also encodes hair color. However, these two alleles may not be identical. One might encode brown hair and the other blonde hair. Okay? So, in the case where the two versions of the gene are identical, we call that homozygosity, meaning that you have identical copies of that bit of information on each chromosome, one from each parent. If they are different, we call that heterozygosity, meaning that you've got different versions of the information on each chromosome. Cells that carry two sets of genetic information are known as diploid cells, and in general, diploidy of the cell indicates how many sets of genetic information it has. The reproductive cells of eukaryotes, like eggs, sperm, and spores, and even non-reproductive cells in some eukaryotes, have a single set of chromosomes and are haploid. The cells of other eukaryotes contain more than two sets of genetic information and are called polyploid. Okay? So a diploid cell, two sets of chromosomes. An example would be, say, an epithelial cell on the surface of your skin. Okay? In the area where the epithelial tissue is being produced. Haploid cell, a sperm cell or an egg cell from a human being would be an example. A polyploid cell, now that's something that you might not have as much familiarity with. Polyploid cell in humans, an example would be a megakaryocyte, okay? But there are also polyploid organisms such as corn, which is hexaploid in many of its, what you'd call its body cells, its vegetative component. Okay? So, an interesting aspect here, right? A homologous pair, again, is the set of chromosomes, one from each parent, that carry similar but not necessarily identical genetic information, while haploid cells carry one set of the genetic information. Okay? Now, when we talk about chromosome structure, the chromosomes of eukaryotes are more complex than prokaryotes. They have unreplicated chromosomes, nevertheless, which consist of a single molecule of DNA, and even though they're linear, the DNA molecules in eukaryotic chromosomes are highly folded and condensed, and if they were stretched out, some human chromosomes would be several centimeters long, thousands of times as long as the span of a typical nucleus. To package all this information into a small volume in the nucleus, each DNA molecule is coiled around the histone proteins and tightly packed, forming a rod-shaped chromosome. Most of the time, the chromosomes are thin and difficult to observe, but during the process of mitosis, they become visible and easily stained. Okay? So you can see here in this panel the 23 sets of chromosomes in the human, right? And the other panel is showing you this idea that we are a diploid organism, right? We have chromosomes that contain similar information from each parent, and the trait, right, that we exhibit is going to be based on the information contained in each of the alleles, okay? As well as, in many cases, information from elsewhere in the genome, okay? All right. One of the reasons that um, we are able to see the chromosomes for the first time during the process of mitosis is because the information has to be protected before we start yanking it all over the cell utilizing centrioles, centrosomes, and microtubules. All right, otherwise it would easily fragment and damage, and the resulting repair might generate mutations. So it's sort of like packing for a trip, right? When you want to take your stuff with you on a trip, you don't just throw it all loose in the trunk of the car. You usually sort it according to type and put it in a suitcase or a satchel, right? And then you put that in the trunk and off you go. And that way you don't damage or lose those items, okay? So this is the idea. Note that a functional chromosome is going to have some essential parts. It'll have a centromere, which is an attachment point for spindle microtubules. It appears as a constricted region on the chromosome. It serves as the attachment point for the spindle apparatus, and these are the filaments that are responsible for moving the chromosomes during cell division. Before cell division, a multi-protein complex called the kinetochore assembles on the centromere, and then spindle microtubules attach to the kinetochore. Chromosomes that lack a centromere cannot be drawn into the newly formed nuclei. These chromosomes are lost, often with disastrous consequences for the cell. On the basis of the location of the centromere, chromosomes can be classed by 4 types: metacentric, submetacentric, acrocentric, and telocentric. Okay? So you can see here, right, some of the different features. The centromere is the attachment point for the spindle microtubules. The telomeres are the tips of linear chromosomes, and the origins of replication, of course, are the regions where DNA synthesis is going to be initiated. Okay? So very critical that we understand that, right? So you can see there the components of a typical chromosome. Keep in mind that when the chromosome looks like this, the information here is the same as the information here. Okay, this is a duplicated chromosome, a replicated chromosome. This is a non-replicated chromosome. Okay, you can see how the duplicated chromosome is held together at the centromere and how the microtubules attach to the kinetochore. And here you can see an example of the karyotype showing you some of the different classifications of chromosomes. Now, the way that the karyotype is done is that cells that are grown in a plastic chamber called a petri dish under conditions that support tissue culture, okay, these will be eukaryotic cells, are frozen at metaphase using a drug called colchicine, okay? Those cells are then osmotically shocked, they rupture, they release their chromosomes. Their chromosomes are then stained and photographed, okay? And the stain is usually a fluorescent stain that produces a banding pattern on the arms of the chromosome. And then we can take that picture and then utilizing programs like Photoshop, but more sophisticated, we can arrange the chromosomes according to their banding pattern so that we can see all of the homologous pairs. Okay, so that is that's the essence of karyotyping. Now, the reason that we worry about karyotyping is because in organisms, if the karyotype is made up of an improper number of chromosomes, an atypical number, that can have disastrous impacts on development. As an example, okay, individuals, humans that have 3 chromosome 21s at the time of fertilization, when the egg meets the sperm in the female reproductive tract, end up with a condition called Down syndrome, in which you have short stature, you have epicanthal folds around the eyes, you have cognitive deficit, you have cardiovascular problems, and you usually have a shortened lifespan, okay? And that's all because the gene dosage in those individuals at the time of fertilization is incorrect. Even though chromosome 21 is one of the smallest chromosomes, there's critical information in there that affects almost every body system. Okay? We had talked about telomeres earlier. Okay? We can hop back for a second and look. These are DNA sequences and associated proteins at the tips of the linear chromosome. These tips protect the genetic information in the chromosome. If a chromosome breaks, producing new ends, the chromosome will be degraded at the newly broken ends, so the telomeres provide stability. We've done research that shows that the telomeres also participate in limiting cell division and may play an important role in aging and cancer. Okay, as an example, right? We have seen that in organisms that have short lifespans, that the chromosomes will shorten with each round of cell division over the lifespan of that organism, and that's because the enzyme that maintains the telomeres, called telomerase, doesn't keep up with the rate of cell division in the lifetime of the organism, usually because the telomerase expression declines as we age, and it's sort of our molecular clock. And then when we get into regions of the chromosome that are critical to life, the cell can't utilize that information anymore, and it commits suicide, and that's a process known as apoptosis. And so as cell death rate accelerates over cell reproduction, eventually cells die, tissues die, organs die, and eventually the organism dies. We've also seen premature aging in conditions in which the DNA structure is prematurely damaged. Progeria is an example of an early aging disease that results from an improperly made protein that's designed to maintain the structure of the DNA in the nucleus. And so what happens when the structure isn't properly maintained is it gets damaged, it gets broken down, that information begins to vanish, and cell death rate accelerates. A lot of people born with progeria seldom make it through puberty. Okay. Origins of replication are the sites where DNA synthesis starts, and unlike centromeres or telomeres, they're not easily observed under the microscope. In preparation for cell division, each chromosome replicates and makes a copy of itself, as already pointed out. The two initially identical copies, known as sister chromatids, are held together at the centromere. Okay? So, there we are. There's our characterization of different chromosomes, right? And now what we want to address is the cell cycle, right? This is a series of stages through which the cell passes from one cell division to the next, and it's through the cell cycle that genetic instructions are passed from one cell to the next. The cell cycle takes place when the cells are actively dividing. A new cycle begins after a cell is divided and produces two new cells. Each new cell grows, develops, and carries out functions specific to its type, and at the end of the cycle, the cell divides and produces two new cells, which undergo additional cycles. Progression through the cycle is regulated at points known as checkpoints, and these allow or inhibit the ability of the cell to progress to the next stage. Checkpoints make certain that cellular components are present in good working order, and these checkpoints are necessary to prevent cells with damaged or missing chromosomes from reproducing. Sometimes defects in checkpoints can lead to conditions such as cancer, which is unregulated cell growth at the expense of the organism that can eventually result in the death of the organism. Okay? When we talk about the cell cycle, we refer to specific parts in the life cycle of the cell, specific times in the life cycle of the cell. Interphase is the time during which the cell is doing its job, okay? An extended period between cell divisions. There's three parts to interphase, G1, S, and G2, as you'll find out shortly. The G1 phases are called gap 1 and gap 2. Gap 1 comes right after mitosis, and gap 2 comes right before it, and synthesis is right before gap 2. Okay, and then there's a period of time in interphase when the cell is simply doing its job. Okay, um, an important point to realize about the cell cycle is that depending on the type of tissue you're talking about, in a multicellular organism, sometimes the cell cycle goes on all the time, such as in an epithelial cell in the skin. Sometimes the cell cycle ceases after birth, such as in neurons in the central nervous system. Okay, so um, that again is dictated by the types of proteins that are present in those particular cells in those tissues that govern when and where the cell cycle will execute. Okay? M phase is the mitotic phase, and these phase checkpoints are critical in determining when and where the cell cycle takes place. Note that cytokinesis is the process by which the cytoplasm of the cell divides, and that usually follows mitosis, where the nuclear material divides. Okay, so it's usually interphase, mitosis, then cytokinesis, and then another round of interphase. Okay, so let's take a look at the cell cycle in more detail.
The two major phases of the cell cycle are interphase, which is the period between cell divisions, and M phase, which is the period of active cell division. Interphase is divided into three phases, G1, S, and G2. A cell may exit from the active cell cycle into a non-dividing phase called G0. M phase is composed of mitosis and cytokinesis. Let's follow a cell with 4 chromosomes through a complete cell cycle, beginning at G1. During G1, the cell grows and proteins necessary for cell division are synthesized. After G1, the cell enters the S phase, in which each chromosome duplicates, so that after cell division, both cells will contain a complete set of genes. At this point, the number of DNA molecules in the cell doubles, but the number of chromosomes remains the same. In G2, several additional biochemical events necessary for cell division take place. At the end of G2, the cell is ready to divide. M phase can be divided into several stages. In prophase, the chromosomes condense and the mitotic spindle begins to form between the centrosomes. Each chromosome consists of 2 sister chromatids joined by 1 centromere. The sister chromatids are 2 identical DNA molecules resulting from the chromosome duplication in S phase. At this stage in the cycle, the cell has 4 functional centromeres and 8 chromatids. In prometaphase, the nuclear envelope breaks down and the spindle microtubules attach to the chromosomes. In metaphase, the chromosomes arrange themselves on the imaginary metaphase plate. In anaphase, sister chromatids separate, becoming individual chromosomes that migrate towards spindle poles. Telophase begins as the chromosomes arrive at the spindle poles. The nuclear envelope reforms and the chromosomes relax. In many cells, cytokinesis takes place simultaneously with telophase. Notice that the number of chromosomes is the same at the end and at the beginning of the cycle. The cell is now ready to begin the cycle anew. What happens during the cell cycle if a cell fails to complete a task, such as DNA replication? If the cell fails to replicate its DNA during S phase, the cell is not ready to divide. The cell cycle will stop at a key transition point called the G2M checkpoint and will not enter mitosis. Let's look at another example. In this case, the cell proceeds through G1, S, and G2 normally. However, a rare failure in prometaphase can occur if the spindle microtubules fail to attach to a chromosome. In metaphase, the spindle fails to arrange the unattached chromosome on the imaginary metaphase plate. If mitosis is not stopped by a checkpoint, then the sister chromatids of the unattached chromosome will not separate or migrate toward the spindle poles in anaphase. In telophase, the nuclear envelope does not form around the unattached chromosome, causing the unattached chromosome to be lost. In another example, the cell proceeds through G1, S, and G2 normally. However, a rare failure in anaphase can occur if the sister chromatids of the chromosome fail to separate. If mitosis continues, then the chromosomes arrive at the spindle poles in telophase. If sister chromatids fail to separate in anaphase, then one cell will have an extra chromatid, whereas the other cell will be missing a chromosome.
So as we can see from what we just watched, interphase is an extended period of growth and development between cell divisions, and even though it appears as if the cell is resting, the cell is actually performing its normal task within the confines of the organism. Interphase is divided into G1, S, and G2 phases. That stands for gap 1, synthesis, and gap 2 phases. Interphase begins with gap 1. In G1, the cell grows and generates proteins necessary for cell division. This stage typically lasts several hours. Near the end of G1, a checkpoint, termed the G1S checkpoint, holds the cell in G1 until the cell has all the enzymes and proteins needed for replication of DNA. After this checkpoint's been passed, the cell is committed to division. Before reaching the G1S checkpoint, the cells can exit the active cell cycle in response to extracellular signals and pass into a non-dividing phase known as G0, which is a stable state during which the cell maintains a constant size, and they can remain in G0 for any amount of time. Okay, in humans, for instance, nerve cells in the central nervous system remain in G0 for the lifetime of the individual to a large extent, whereas an epithelial cell won't remain in G0 for very long at all. Okay? It all depends on the role that that cell plays within the context of that tissue, in the context of that organism. Okay? After G1, the cell then enters S phase, during which DNA is synthesized. Although the cell is committed to divide after the G1S checkpoint is passed, DNA synthesis has to take place before the cell proceeds into mitosis. If DNA synthesis is blocked, the cell will not normally be able to undergo mitosis. Before S phase, each chromosome is unreplicated, and after S phase, they're made up of two sister chromatids, which won't become visible until prophase of mitosis. After S phase, we enter G2. In this stage, additional biochemical events important for cell division take place. The G2M checkpoint is reached near the end of G2, and it's passed only if the cell's DNA is completely reproduced and undamaged. Unreplicated or damaged DNA can inhibit the activation of proteins necessary for mitosis. After this checkpoint is passed, the cell is ready to divide and enters the M phase. Although the length of the interphase varies from cell type to cell type, a typical dividing mammalian cell spends about 10 hours in G1, 9 in synthesis, and 4 in G2. Although interphase indicates that the chromosomes are in a relaxed state, while still associated with their histones and their nuclear proteins, they are by no means uncoiled, and individual chromosomes can't be seen with a microscope. This condition changes dramatically when interphase draws to a close and the cell enters M phase. And so what happens here is that those chromosomes are going to condense and become visible for the first time. So it's during M phase that we see the chromosomes, okay? And of course, M phase is divided into prophase, anaphase, metaphase, and telophase, right? Here we see the critical points for interphase that we just discussed. M phase results in separation of the sister chromatids, and that's generally followed by cytokinesis, which is the separation of the rest of the cellular components. M phase begins with prophase. As the cell enters this stage, the chromosomes condense and become more compact and visible under a light microscope. Proteins called condensins bind to chromosomes and bring about this process. Because the chromosomes were duplicated in the preceding S phase, each chromosome has two sister chromatids attached at the centromere, and the genetic information on each arm of the chromosome on sister chromatids is identical, unless there's been a mutation. The mitotic spindle, which is an array of microtubules that move the chromosomes in mitosis, forms, and in animal cells the spindle grows out from a pair of centrosomes that migrate to the opposite ends of the cell. Within each centrosome there's an organelle called the centriole, which is also made of microtubules, although in plant cells we do not see mitotic spindles. Condensins are a group of proteins that bind chromosomes as the cell enters prophase and causes the chromosomes to become more compact and visible under the light microscope, and prophase is the first stage of mitosis during which the chromosomes are visible. Okay? Prometaphase sees the disintegration of the nuclear membrane and begins essentially the dispersion of the chromosomes. Spindle microtubules, which had been outside the nucleus, enter the nuclear region and attach to the chromosomes. During prometaphase, tubulin molecules are added and removed from the microtubules, and they undergo repeated cycles of growth and shrinkage as a result. When the end of a microtubule hits a kinetochore, the microtubule is stabilized. Eventually, each chromosome becomes attached to microtubules from opposite spindle poles, and what will happen when force is applied across the centromere is that the duplicated chromosomes will be separated. Okay? In metaphase, the chromosomes become arranged in a single plane called the metaphase plate, which is between the two centrosomes. It's not a physical structure, but basically a location. The centrosomes, which are at opposite ends of the cell at this point, have microtubules radiating outward, each one meeting in the middle of the cell, which has the center at the spindle poles. A spindle pole assembly checkpoint ensures that each chromosome is aligned on the metaphase plate and attached to the spindle microtubules from opposite poles. And then in metaphase, we see the chromosomes, once aligned, prepare for separation. What happens as metaphase transitions into anaphase is that tension is produced at the kinetochore as the two conjoined chromatids are pulled in opposite direction by spindle microtubules. The tension is necessary for the cells to pass through this checkpoint. If one chromatid is attached to a microtubule but the other is not, no tension is produced and the cell cannot go to the next stage of mitosis. The spindle assembly checkpoint can detect even a single pair of chromosomes that aren't properly attached to microtubule sets. The importance of this checkpoint is illustrated by cells that are defective in their spindle assembly. These cells often end up with abnormal numbers of chromosomes in the daughter cells. Okay? So you can see these steps here, right? Illustrated. Okay? The 6 stages of mitosis. Right? And here you can see the microtubules and their tubulin subunits. We have to appreciate the fact that this is a hollow assembly which is built and broken down by the centrosome, and the tubulin subunits are always present in the cytoplasm, ready to be polymerized or taken apart. Okay? We should also note that the microtubules are also present throughout the cell's life cycle, but when we're not undergoing mitosis, the role of microtubules is to serve as a mechanism for manipulating the positions of organelles in the cell so that they can perform at their optimal efficiency. And it also aids in the movement of transport and secretory vesicles. Okay? In anaphase, the spindle assembly checkpoint has been passed. The connection between the sister chromatids breaks down. Okay? And the duplicated chromosomes are going to separate and move towards opposite spindle poles. Special proteins called molecular motors disassemble tubulin molecules and generate forces that pull the chromosome towards the pole. Okay? A condition known as amphidiploidy can occur. It's a type of allopolyploidy in which two different diploid genomes are combined such that every chromosome has one and only one homologous partner, and the genome is functionally diploid. Okay? So we can see the different phases here outlined in the table. Okay? In telophase, the sister chromatids have separated. Each is considered a separate chromosome at this point. The chromosomes arrive at the spindle poles. The nuclear membrane reforms around each set of chromosomes, generating separate nuclei in the cell. The chromosomes relax and lengthen and become no longer visible under a light microscope. In most cells, division of the cytoplasm occurs during telophase as well. The major features of the cell cycle were summarized in the previous video. Okay? The consequences of the cell cycle are that we generate two daughter cells that are genetically identical to each other unless there's been a mutation. Okay? The new cells have a full complement of genetic information and has about half the cytoplasm and organelle content of the original parent. Okay? So very important that we understand how this works. You can see here the process, right? We're moving through the cell cycle in this diagram. The number of chromosomes per cell equals the number of functional centromeres. The number of DNA molecules per cell equals the number of chromosomes when the chromosomes are unreplicated, i.e. no sister chromatids are present, and twice the number of chromosomes when the sister chromatids are present. Okay, so that's how we do the bookkeeping here. Okay. Okay. Next topic we want to address is meiosis. Okay. One of the things we have to appreciate about cell division is that there are two methods of generating new organisms, okay? Sexual and asexual reproduction. We've seen asexual reproduction when we looked at binary fission in bacteria and mitosis in eukaryotic cells, okay? What happens at the end of the process is that you get two daughter cells that have the same genetic complement and are going to look and act like the original cell with very few exceptions. Okay. The upside of this is it's a rapid process. In sexually reproducing organisms, mitosis is for growth, maintenance, and repair of tissue. Okay. In asexually reproducing organisms, mitosis is not only for growth, maintenance, and repair, but also for reproduction. The downside of this is that every organism is identical to the original unless there is mutation. The result is that if the organism encounters an environmental challenge that sickens or kills one member of the population, it's likely that the entire population will get wiped out. Okay? So the way that mitotically reproducing organisms deal with environmental challenge is by sheer numbers. There's a downside to this, which is that the population, because it lacks biodiversity, is limited in its ability to adapt to changing conditions. This is why sexual reproduction evolved during the changes that took place in life on this planet. Okay, in sexual reproduction, there are sex cells, eggs and sperm, that are haploid that come together in a process called fertilization to produce a diploid cell, which is the first new cell of a new organism. And the result of meiosis is that the organism that's generated is not identical to either parent. It's similar, but because it's getting traits from both the male and the female, the resulting organism will have physical, biochemical, and biological differences. And this is going to generate traits upon which selection can act. In addition, we're going to find out that during the course of meiosis, a phenomenon called crossing over, in which homologous pairs, once produced, exchange genetic material between sister chromatids on different chromosomes, results in novel arrangements of genes that aren't present in the body cells of either parent. And that adds to more biodiversity, and as a result, a greater series of characteristics upon which selection can act. Okay? So, the evolution of sexual reproduction is a significant event in the history of life. We're going to talk about it much later in the course. The pace of evolution depends on the amount of genetic variation in the population, and by shuffling information from two parents, sexual reproduction increases the amount of biodiversity and accelerates evolution, which acts on physical expression of characteristics. Most of the tremendous diversity of life on the planet is the result of sexual reproduction. Sexual reproduction has two processes. The first is meiosis, which leads to haploid gametes at half the number of chromosomes that a body cell has. The second process is fertilization, in which the haploid gametes fuse and restore the diploid number in the zygote, which is the first cell of a new individual. Okay? Double fertilization is seen in plants. This is a form of fusion of sperm and egg cells that generates a zygote, and a second sperm cell fuses with polar nuclei and generates something called endosperm, which is primarily for the protection and the distribution of the resulting zygote. Okay? And of course, meiosis we've discussed, right? There's two rounds of meiosis in every meiotic division. There's meiosis 1 and meiosis 2. Meiosis 1 is referred to as the reductional division because it reduces by half the number of chromosomes in the resulting cell. And meiosis 2 is the equational division because the 4 cells that result have the same number of chromosomes as the parent cell from meiosis 1, but half the number of chromosomes of the original diploid cell. Okay? And in addition, there are novel genetic arrangements on those resulting chromosomes. Note that mitosis and meiosis can be confused. They sound a lot alike, but they can appear superficially similar, but actually there are important differences. Mitosis, remember, is a single nuclear division accompanied by a single round of cytokinesis most of the time. Meiosis, on the other hand, is 2 rounds of cell division. After mitosis, the chromosome number in the newly formed cells is the same as in the parent, but after 2 rounds of meiosis, we generate 4 cells that have half the amount of genetic material as the original cell. Like mitosis, meiosis is preceded by an interphase that has a G1S and a G2 component. Meiosis has 2 distinct processes, meiosis 1 and 2, which we've previously discussed. Okay? So you can see here in meiosis, right, the important phases, right? DNA synthesis and chromosome replication, and interphased, and meiosis I, the reductional division, and meiosis II, the equational division, and the resulting products shown here. Okay, 2 cell divisions. All right. The reason that this happens is because in the transition from meiosis I to meiosis II, there is no duplication of DNA that takes place. So even though this appears a lot like an interphase, it isn't. Okay, the cell DNA content is not changing during this period of time, and then we undergo the equational division, and as you can see, each of these 4 cells with 2 chromosomes had half the amount of chromosomes as the original cell, which had 4. Okay, remember, these are duplicated chromosomes, and these are homologous pairs. Right? Meiosis I is preceded by an interphase. The chromosomes are relaxed but indistinguishable, and they appear as diffuse chromatin. Prophase I is a lengthy stage divided into 5 substages. In leptotene, the chromosomes condense and become visible. In zygotene, the chromosomes continue to condense. Homologous chromosomes pair up and begin synapsis, where they pair with each other extremely closely. Each homologous pair of synapse chromosomes forms what we call a bivalent or a tetrad, which consists of 4 chromatids. In pachyotene, the chromosomes become shorter and thicker, and a 3-part synaptonemal complex develops between homologous chromosomes. The function of the synaptonemal complex is unclear, but the chromosomes of many cells deficient in this complex don't properly separate. Okay? So we see, again, some important terminology that we need to know, right? Now, crossing over is a critical event that happens. Crossing over occurs when homologous chromosomes exchange genetic information. This takes place in prophase I. Crossing over generates genetic variation and is essential for the proper alignment and separation of homologous chromosomes. Each location where 2 chromosomes cross is called a chiasmata, or a chiasma. The plural is chiasmata. In diplotene, the centromeres of the paired chromosomes move apart, but the 2 homologs are attached at the chiasma. Near the end of prophase I, the nuclear membrane breaks down and the spindle forms, and that sets the stage for metaphase I. The stages of meiosis are outlined in the figure here. Okay, you can see here the equational and the reductional division. Okay, and then you can see here in more detail, right, what happens during the components of prophase. Okay, so these are each parts of prophase. All right. Now, an important point is that crossing over is going to create haploid cells with unique arrangements of chromosomes, and what determines where a crossover will take place isn't completely clear. Okay, we do know that there are regions in certain chromosomes that are crossover hotspots, and so recombination or exchange of genetic material happens at a greater rate in these regions than it does in other places on the chromosome. And this is one of the things that can confuse what we call genetic distance with physical distance in terms of the amount of distance on the chromosome that separates 2 genes. Okay, genetic distance is measured by the degree of recombination that takes place between 2 sets of alleles in an organism. Okay, the greater the degree of recombination, the greater the genetic distance between those 2 genes on a particular chromosome. Whereas physical distance is nothing more than the separation along the chromosome in terms of DNA sequence. Okay, so we have to make this distinction here because we have to be aware of the fact that the tendency to cross over is not uniform along the entire length of the chromosome. Metaphase 1 is initiated when homologous pairs of chromosomes align along the metaphase plate. A microtubule from one spindle pole attaches to one chromosome of the homologous pair, and a microtubule from the other pole attaches to the other member of the pair. Anaphase 1 is marked by the separation of homologs. The two chromosomes of a homologous pair pull towards opposite poles. Although the homologs separate, the sister chromatids are still attached and travel together. In telophase 1, the chromosomes arrive at the spindle pole and the cytoplasm divides. And the result of that is a reductional division. We generate two haploid cells. Okay? Some other important players here. Okay? Aminoacyl tRNA synthetase is an enzyme that attaches an amino acid to a transfer RNA. Each aminoacyl tRNA synthetase is specific for a particular amino acid and is an important component in the process known as translation. Okay? Metaphase 1 is a stage of meiosis in which the homologous pairs of chromosomes align in the center of the cell. Okay? So you can see here that this is going to be followed by meiosis 2. Okay? Meiosis 2 is going to follow interkinesis, which is the period between meiosis 1 and 2. During interkinesis, remember that no duplication of genetic material takes place. The nuclear membrane reforms around the chromosomes clustered at the pole. The spindle breaks down and the chromosomes relax. The cell then passes through prophase 2, in which the events of interkinesis are reversed. The chromosomes recondense, the spindle reforms, and the nuclear envelope once again breaks down. In interkinesis, in some types of cells, the chromosomes remain condensed and do not unwind, and the spindle apparatus doesn't break down. These cells move directly from cytokinesis into metaphase 2, which is similar to the metaphase of mitosis. The replicated chromosomes line up along the metaphase plate with their sister chromatids facing opposite poles, but the orientation of those duplicated chromosomes is random. Okay, so they're in that particular event. Okay, the orientation in metaphase 1 of the homologous pairs and the orientation at metaphase 2 of the duplicated chromosomes means that there is going to be independent assortment of characteristics that are carried on different chromosomes. Okay, so those chunks of DNA will be pulled into a gamete, all right, but there is no rule that says that a particular chromosome, say number 1, has to follow a particular chromosome number 2, and so on. Okay, so this further increases biodiversity as a result. Okay, in anaphase 2, sister chromatids separate and the chromatids are pulled to opposite poles. Each chromatid is now a distinct chromosome, and in telophase 2, the chromosomes arrive at the spindle poles, the nuclear envelope reforms, the cytoplasm divides, the chromosomes relax and are no longer visible. Telophase 2 is the stage of mitosis 2 where the chromosomes arrive at the spindle poles. Anaphase 2, the stage of meiosis 2, in which the chromatids separate and move towards the spindle poles. Okay, and again, you can see the entire process here. Okay, and you can see the important components here. All right, these tables and these graphics are important. I would encourage you to familiarize yourself with what each of these phases looks like and the definition of the critical events at each phase. Okay? So what are the consequences of meiosis? Well, we generate 4 cells from each original cell. The chromosome number is now reduced by half, but the newly formed cells are genetically different from one another and from the parents. Now, why is this? Well, 2 reasons, right? the orientation of homologous pairs and duplicated chromosomes at metaphase meiosis 1 and 2 respectively, and the crossing over that takes place during prophase meiosis 1, in which genetic material is exchanged, resulting in novel arrangements of genes not present in parental DNA. Okay? And then at fertilization, we increase this even further because now we bring in the DNA of another parent. Okay? So you can see here an example of meiosis illustrated with alleles outlined and the resulting cells that can take place, right? Note that crossing over occurs as a result of breaks in DNA strands. Those breaks are repaired in such a way that segments of non-sister chromatids exchange. The molecular basis will be described later. The important thing to note is that after crossing over has taken place, the sister chromatids are no longer identical, okay? So what you have are novel arrangements of genes that are not present in the diploid cells of that parent, okay? Eventually, the two homologs will separate, each going into a different cell. In meiosis 2, the two chromatids of each chromosome separate, and thus each of the 4 cells resulting from meiosis have a different combination of alleles. You can see how crossing over affects this genetic variation when we view the next animation, okay, which is going to come up here in just a second, okay. We should also note, again, random separation of homologous chromosomes, and this is just pointing out the fact that while the homologous pairs that you get from each parent are similar, all right, the individual that produces the gametes is going to produce homologous pairs from their parental or maternal DNA that don't have to line up in any particular orientation along the metaphase plate at prophase or at metaphase of meiosis 1, okay? And so what happens as a result of this is that you get random assortment and random segregation of these homologous pairs, okay? And then we also pointed out that at metaphase meiosis 2, the orientation of the duplicated chromosome on the metaphase plate is not something that is predetermined. It is a random event, all right? So let's take a look at some aspects of meiosis to make this more clear.
As in mitosis, meiosis is preceded by interphase during which the cell grows, duplicates the chromosomes, and prepares for division. Meiosis consists of 2 phases, meiosis 1 and meiosis 2. Meiosis 1 begins with prophase 1. In this stage, chromosomes condense, the mitotic spindle forms, and the nuclear envelope breaks down. Importantly, homologous chromosomes pair with each other, and crossing over, not shown here, takes place in prophase 1. In metaphase 1, homologous pairs of chromosomes line up on the metaphase plate. In anaphase 1, the homologous pairs separate and move toward opposite spindle poles. In telophase 1, the chromosomes arrive at the spindle poles, and the cytoplasm divides. The result of meiosis 1 is 2 cells, each containing half the original number of chromosomes. The cell then moves into meiosis 2, which is similar to mitosis. The chromosomes line up on the metaphase plate in metaphase 2. The sister chromatids separate in anaphase 2. In telophase 2, the chromosomes arrive at the spindle poles. The nuclear envelope reforms, and the cytoplasm divides. The result of meiosis is 4 cells, each containing half the original number of chromosomes. Let's look at a few ways in which rare errors can occur in meiosis I. Consider what happens if a set of homologous chromosomes fail to pair in prophase I and the cell continues through meiosis. In this case, at the end of meiosis I, one cell will contain an extra chromosome and the other cell will be missing a chromosome. The cells then proceed through meiosis II. How do these cells compare with cells that have undergone a normal round of meiosis? As another example, consider what happens if non-homologous chromosomes pair in prophase I and the cell continues through meiosis. In this case, each cell will contain a different set of chromosomes at the end of meiosis I. The cells then proceed through meiosis II. Note that each cell contains two homologs of a particular chromosome and lacks a copy of the other chromosome. How do these cells compare with cells that have undergone a normal round of meiosis? Now consider an example in which the correct chromosomes pair with each other, but these pairs of homologous chromosomes fail to separate during anaphase I. At the end of meiosis I, the resulting cells each contain a different set of chromosomes. The cells proceed through meiosis II. Each of the resulting cells has an incorrect number of chromosomes.
Crossing over during meiosis allows recombination of genes between homologous chromosomes. This alters the linkage between genes on the same chromosome. Crossing over occurs frequently during prophase I of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for two genes on a particular chromosome when crossing over does not occur. Recall that one member of each pair of homologous chromosomes came from one parent. The other member came from the other parent. In this example, one parental chromosome had alleles capital A and capital B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of linked genes is not altered in this case. Now consider the case where there is crossing over in the region between the two genes. Since the crossing over is only between two individual chromatids, the possible haploid cells after meiosis have four different genetic combinations. Two will come from the chromatids that did not participate in crossing over and thus will still have the parental combination of alleles, either capital A and capital B, or lowercase a and lowercase b. The other two will be recombinant, with either capital A and lowercase b, or lowercase a and capital B alleles. Thus, crossing over allows for the reassortment of linked genes.
So you can see in this diagram the events of meiosis is outlined in the previous two video slots, okay? And we can see here, all right, the consequences of each event in each process, right? Mitosis, meiosis 1, and meiosis 2. Okay. In recent years, some of the molecules required for the joining and separation of chromatids and homologous chromosomes have been identified. Cohesin is a protein that holds chromatids together and is important in the behavior of chromosomes in both mitosis and meiosis. The sister chromatids are held together by cohesin, which is established in the S phase and persists through G2 and early mitosis. In anaphase of mitosis, cohesin along the entire length of the chromosome is broken down by an enzyme called separase, and that allows the sister chromatids to separate. So cohesin is a major player here. All right. If we look at the mechanism, right, you can see how the cohesin is holding the genetic material together in mitosis and meiosis. All right. But something we need to point out, all right, is that when we're talking about the generation of homologous pairs, how is it that a homolog can find its mate? Okay, and the answer comes from DNA sequence. All right, the closer the DNA sequence matches on a particular stretch of chromosome, the more likely they are to pair up, and that's why the homologs tend to find each other based primarily on the DNA sequence. The bottom line is that while the mechanism remains somewhat elusive, it is the fact that there is similar information along the DNA stretches of the chromatids that is an important factor in them aligning together to form the tetrad. Okay, we know this because there are events such as translocations where the arm of one chromosome ends up on a different chromosome that result in the formation of abnormal chromosomal conformations, okay, that are obviously the result of trying to match homologous sequence, okay, and that usually results in aneuploidy and the resulting meiotic products, okay. As we've seen, mitosis and meiosis differ in the behavior of the chromosomes in anaphase. Why do homologs separate in anaphase of meiosis, but chromatids separate in anaphase of mitosis and anaphase 2 of meiosis? It's important to note that the forms of cohesin used in mitosis and meiosis are different. At the beginning of meiosis, the meiosis-specific cohesin is found along the entire length of the chromosome arms. The cohesin also acts on the chromosome's arms of homologs at the chiasmata, which are the crossover points between homologous chromosomes that tethers the homologs together. In anaphase 1, cohesin along the chromosome arms is broken, allowing the two homologs to separate. However, the cohesin at the centromere is protected by a protein called shugosin. Because of the protective action of this protein, the centromere cohesin remains intact and prevents the separation of the two sister chromatids during anaphase 1. Shugosin is subsequently broken down. At the end of metaphase 2, the centromeric cohesin, no longer protected by shugosin, breaks down and allows the sister chromatids to separate in anaphase 2, just like they do in mitosis. Okay? The overall result of meiosis is 4 haploid cells that are genetically variable. Meiosis fits into the life cycles of multicellular animals and plants in slightly different ways. Okay? If we look at meiosis in animals, we have 2 processes, spermatogenesis and oogenesis. Spermatogenesis is male gamete production. Oogenesis is female gamete production. And then we have meiosis in plants. Okay? So, in spermatogenesis, all right, the testes are the site of sperm production. Primordial germ cells called spermatogonia divide mitotically and then enter meiosis, generating the meiosis I products, the primary spermatocytes, which are haploid. The homologous chromosomes have not yet separated. Each primary spermatocyte completes meiosis I, giving rise to two haploid secondary spermatocytes that undergo meiosis II, with each producing two haploid spermatids. Thus, each primary spermatocyte produces a total of 4 haploid spermatids, which mature and develop into sperm. Okay? So, once we're through prophase 1, the primary spermatocyte, a diploid cell, is now genetically distinct from the spermatogonia that preceded it because crossing over has taken place. So it's a diploid cell, but the gene's been rearranged. At the beginning of meiosis II, okay, the secondary spermatocytes are going to undergo the equational division and generate 4 haploid gametes that are going to be unique in their genetic composition. Okay? The spermatogonia, remember, is the diploid cell in the testis that undergoes division to produce a sperm. Spermatogenesis is sperm production in animals. The primary spermatocyte is the spermatogonia that's entered prophase 1 of meiosis. The secondary spermatocyte is the product of meiosis I in the male, and the spermatid is basically the physically modified secondary spermatocyte that will eventually mature into a sperm cell. Okay? In the female, we have a slightly different situation. Okay? Oogenesis occurs as a result of oogonia dividing mitotically. Okay? Like spermatogonia, oogonia undergo repeated rounds of mitosis. They then enter prophase 1, and these diploid cells, known as primary oocytes, are going to essentially pause in the ovary. Right? Each primary oocyte completes meiosis I and divides. So the primary oocyte is the oogonia that's entered prophase 1, and oogenesis is just the egg production that takes place. At this point, oogenesis begins to differ from spermatogenesis. In oogenesis, cytokinesis is unequal. The cytoplasm allocation in the first and second divisions produces polar bodies and oocytes. Okay? Most of the cytoplasm is allocated to one of the 2 haploid cells, the secondary oocyte, and the smaller cell, which has only a small amount of cytoplasm, the first polar body, may not divide further. The secondary oocyte completes meiosis II, and again, the cytokinesis is unequal. Most of the cytoplasm passes into one of the cells. The larger cell acquires most of the cytoplasm, and that is the ovum, which is the mature female gamete. The second smaller cell is the second polar body. Only the ovum is capable of being fertilized, and the polar bodies usually disintegrate. Okay? So let's review this process.
Spermatogonia are the cells from which sperm cells arise. The spermatogonia divide by mitosis. One daughter cell remains a spermatogonium, and the other becomes a primary spermatocyte. The primary spermatocyte divides by meiosis to form secondary spermatocytes. Secondary spermatocytes divide again to form spermatids. The spermatids differentiate into sperm cells.
In prophase 1, the DNA coils tightly and individual chromosomes become visible under the light microscope. Homologous chromosomes become closely associated in synapses and they exchange segments by crossing over. By metaphase 1, the nuclear membrane has disappeared and the microtubules form a spindle. Spindle fibers attach to only one side of each centromere and the two homologous chromosomes attach to microtubules orienting from opposite poles. Each pair of homologs then lines up on the metaphase plate. Either maternal or paternal homolog may orient toward a given pole. In anaphase 1, the microtubules of the spindle fiber shorten and pull the chromosomes toward the poles, taking both sister chromatids with them. Each pole ends up with a complete haploid set of chromosomes consisting of one member of the homologous pair. During telophase 1, the nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains two sister chromatids for each chromosome attached to a common centromere. Because of crossing over, the two sister chromatids are not identical. During prophase 2, the nuclear envelope breaks down and a new spindle forms. In metaphase 2, spindle fibers bind to both sides of the centromeres. During anaphase 2, the spindle fibers contract and the sister chromatids move toward opposite poles. In telophase 2, nuclear envelopes reform around the sets of daughter chromosomes.
Spermatogonia are the cells from which sperm cells arise. The spermatogonia divide by mitosis. One daughter cell remains a spermatogonium, and the other becomes a primary spermatocyte. The primary spermatocyte divides by meiosis to form secondary spermatocytes. Secondary spermatocytes divide again to form spermatids. The spermatids differentiate into sperm cells.
In human females, oogenesis, or egg formation, takes place within the ovaries. Each ovary contains diploid cells called oogonia, derived from embryonic germ cells. Before a woman's birth, the oogonia divide by mitosis. The result is more oogonia, some of which develop into primary oocytes. Primary oocytes are immature egg cells contained within masses of cells called follicles. The primary oocytes then enter meiosis I. This process stops uncompleted until puberty. No primary oocytes are formed after this point. At birth, each female has a finite number of primary oocytes available for reproduction. At puberty, a complex series of hormonal events stimulates changes in the surrounding follicle and induces some primary oocytes to complete their first meiotic division. The division of cytoplasm and cell organelles is unequal, however. As a result, one large secondary oocyte and one small polar body form per primary oocyte. The polar body often degenerates. The follicle containing the secondary oocyte continues to mature until a surge of the hormone LH initiates ovulation. The mature follicle ruptures, releasing the secondary oocyte. At this point, the secondary oocyte has entered meiosis II. This second meiotic division will not be completed, however, unless fertilization occurs. The secondary oocyte enters the oviduct, where fertilization of the oocyte with a sperm cell can occur. The entry of a sperm cell into the cytoplasm of the secondary oocyte triggers the completion of meiosis II. The cytoplasm divides unequally, generating a mature ovum and a second polar body. The fusion of the haploid sperm cell and the haploid egg cell has produced a diploid zygote. Let's compare spermatogenesis with oogenesis. Both processes start before birth, when embryonic germ cells differentiate into spermatogonia in the testis of a male, or oogonia in the ovary of a female. Both types of cells are diploid. However, whereas mitotic divisions continue to generate new spermatogonia in the male until death, in the female, the generation of more oogonia by mitosis halts well before birth. Although spermatogonia develop into primary spermatocytes throughout life in the male, in the female some oogonia develop into primary oocytes, but only before birth. Males can continue to produce viable sperm from puberty until death, but females can produce viable eggs only from puberty until the supply of primary oocytes is depleted. Meiosis I does not begin until puberty in the testis of the male. Meiosis I begins before birth and ends at puberty in the ovary of the female. Meiosis II occurs within the testis at any time from puberty until death in the male. Meiosis II begins within the ovary only at ovulation in the female and ends within the oviduct upon fertilization by a sperm cell. The final result of spermatogenesis is 4 haploid sperm cells for each spermatogonium. In contrast, oogenesis can result in 1 egg cell and 2 polar bodies for each oogonium, but only if fertilization has occurred.
Now in plants, the process differs yet again. Most multicellular plants and algae have a complex life cycle that involves 2 structures, a multicellular diploid sporophyte and a multicellular haploid gametophyte. These 2 generations alternate. The sporophyte produces the haploid spores through meiosis, and the gametophyte produces the haploid gametes through mitosis. This type of life cycle is sometimes called alternation of generations. In this cycle, the immediate product of meiosis are called spores, not gametes. The spores undergo 1 or more mitotic divisions to produce the gametes, although the terms used to describe plant life and the plant life cycle are a little different from those used in animals. The process in plants and animals is basically the same. In both, meiosis leads to a reduction in chromosome number and produces haploid cells and increases genetic variability. In flowering plants, the sporophyte is the obvious vegetative plant. The gametophyte consists only of a few haploid cells within the sporophyte. The flower, which is part of the sporophyte, contains the reproductive structures. In some species, both male and female reproductive structures are found in the same flower. In other species, they're in different flowers. In either case, the male part of the flower, known as the stamen, has the diploid reproductive cells called microsporocytes. Each undergoes meiosis to produce 4 haploid microspores. Each microspore divides mitotically to produce immature pollen grains that have 2 haploid nuclei. One of these nuclei, called the tube nucleus, directs the growth of a pollen tube. The other, termed the generative nucleus, divides mitotically to produce the sperm cells. The pollen grain, with its haploid nuclei, is the male gametophyte. So again, to review, the microsporocyte is the diploid reproductive cell in the stamen of the plant that undergoes meiosis to produce the haploid microspores, which are the haploid product of meiosis. The female part of the flower, which is the ovary, contains diploid cells called megasporiocytes, each of which undergoes meiosis to produce 4 haploid megaspores, only one of which will survive. The nucleus of the surviving megaspore divides mitotically 3 times, producing a total of 8 haploid nuclei that make up the female gametophyte, otherwise known as the embryo sac. Division of the cytoplasm produces separate cells, one of which becomes the egg. The megaspore is one of the 4 products of meiosis in plants, while the megasporocyte is a diploid reproductive cell in the ovary of the plant that undergoes meiosis to generate the haploid macrospores. When the plant flowers, the stamens open and release the pollen grains, which land on the sticky stigma, which sits on top of a stalk called the style. At the base of the style is the ovary. If a pollen grain germinates, it grows a tube down the style into the ovary. The two sperm cells pass down this tube and enter the embryo sac. One of the sperm cells fertilizes the egg cell and generates the zygote, which is going to form the embryo, and the other sperm cell fuses with two nuclei enclosed in a single cell, giving rise to a triploid endosperm, which stores food that will be used later by the embryonic plant. These two fertilization events are collectively termed double fertilization. Okay? So you can see the process outlined here and here. Okay? And we will also review it in the upcoming video. Okay? So I will see everybody in the next chapter.
A couple of weeks ago, I talked about a strategy for reproduction that the very first plants came up with called alternation of generations. A strategy that non-vascular plants still use today. Hopefully this is coming back to you. A plant can take two different forms that alternate back and forth between generations. The first form, the sporophyte, has diploid cells, two sets of chromosomes, and the second form, the gametophyte, has haploid cells, just one set of chromosomes. Well, a lot can happen in 470 million years. Today, vascular plants still use the basic alternation of generations model, but they've tricked it out so that it works a whole lot different than it did back in the Ordovician swamps where planthood got its start. Compared with their small, damp, non-vascular brethren, vascular plants with all their cones and flowers and other flashy accessories look like a bunch of drag queens at a Carmen Miranda conference and samba dance-off. Which might seem like overkill, but we rely on these crazy kooks and their upstart reproductive strategies for, well, pretty much for our everything. The food we eat, the air we breathe, the bouquets that we send to our wives and girlfriends when they're mad at us. Basically, what I'm saying is that we need vascular plants to have sex. So as you'll recall, the alternation of generations in non-vascular plants is pretty straightforward. A gametophyte produces either sperm or eggs, which find each other if it's wet enough for the sperm to swim to the nearest egg. Once the egg is fertilized, the gametophyte creates the sporophyte, which is a little capsule on a stalk that has a bunch of spores in it. The spores are released into the air, they land in a moist place, germinate, and bam! A new gametophyte generation is born. But non-vascular plants are what you call gametophyte dominant. What you're looking at when you look at a moss or a hornwort or a liverwort is the gametophyte. It's the form that has only one set of chromosomes. For them, the sporophytes are tiny and tucked away inside the gametophytes, which they rely on for food, water, and protection. But in vascular plants, it's the opposite. They're sporophyte dominant. When you look at a fern or a pine tree or a morning glory, you're looking at the sporophyte generation. And the gametophytes are the teeny tiny sex-making materials it has stashed away in special parts. So yes, all vascular plants are sporophyte dominant, but that does not mean that they all reproduce in the same way. No, sir. The simplest form of vascular plants are the ferns, which reproduce a lot like non-vascular plants in that they have spores that grow on the underside of the frond or fern leaf, which are released into the wild blue yonder to find a nice soggy patch of ground to germinate on. The spore then makes a tiny gametophyte, which is only a few centimeters wide and has both male and female reproductive organs on the underside of its leaves. If it's moist enough, the sperm on the boy side of the gametophyte will find the egg on the girl side, and it will create a sporophyte, which is what we recognize as a fern. There's a lot of fossil evidence to suggest that at one point there probably were ferns that produced seeds, and that all the fancy pants vascular plants that do have seeds and flowers evolved from them. But those seed-bearing ferns are all extinct now, so we can just gaze longingly at their fossils and wonder what their alternation of generations looked like. But there are other groups of plants that are more complex than ferns, and what they all have in common is that they reproduce by creating pollen, which contains the male gametophyte, and the female gametophytes or ovules, which are fertilized by the pollen. The complete fertilized cell grows into a seed, which ripens and can produce a complete adult plant. So, reiterating, in your more advanced vascular plants, that's how the alternation generation works. The sporophyte generation grows from a seed and produces tiny gametophytes, either pollen or ovules. They then combine to form another seed, which produces another sporophyte. This evolutionary change from spores to seeds was a big deal, and it began with the gymnosperms. They're single-serving plant-making packages cut out the middleman by allowing an adult plant to grow immediately from a seed, rather than having to wait for a spore to go through that intermediate gametophyte stage. It also means, in most cases, that there doesn't have to be water present in order to reproduce. Today, gymnosperms include conifers, ginkgos, and tropical palm-like plants called cycads, and none of them produce flowers because they evolved before flowers were invented. Instead, their reproductive structures are cones, and you've seen a few of these in your day. In fact, their name gymnosperm means naked seed, and that comes from the fact that their ovules develop exposed on the surface of the cone scales. Now, what we think of as cones are the spiky woody things that Boy Scouts are throwing at each other at camp, right? But those things are actually female cones which house the ovules. The male cones are smaller and kind of spongy, and their job is to crank out pollen. All this pollen is carried on the wind, and some of it might find its way to a female cone where it fertilizes the ovule located at the base of each of the scales of the female cone. As the fertilized embryo matures inside the cone, it makes a seed containing enough nutrients to sustain it for a while after it germinates. This seed has a tough, shiny casing to protect it from the elements, and once it's mature, the scales of the female cone just peel back, and the seed falls to the ground and makes a new tree. But some gymnosperms have evolved the need for special conditions in order to reproduce. Take the lodgepole pine. It's a super tough tree that evolved in a pretty dry climate where there's lots of lightning storms that regularly start fires that burn through a forest every few years. Not only do lodgepoles have no problem withstanding a good low-intensity forest fire, their female cones are serotinous, so they will only open and drop their seeds when exposed to extreme heat. Now, this sounds kind of crazy, but really it's super smart because the lodgepoles have evolved to take advantage of forest fires. They know that the forest fire will probably get rid of a lot of pesky underbrush that would crowd out their babies, and maybe even it would kill some adult lodgepole pines, so they just wait for the competition to be removed before they expose their seeds. So now I'm fixing to pull out the big guns, the angiosperms, because angiosperms are the winners of the all-invitational plant division of things that live on Earth. At least for the past 140 million years or so. They're rookies, really, but they know what they're doing. For starters, they have seeds like gymnosperms, but they also have flowers. And flowers are awesome because they don't have to rely on the wind to carry their pollen to another flower like gymnosperms do with their cones. For the most part, flowers put animals to work toting their pollen from one flower to another. In fact, angiosperms and flying insects probably evolved together, or co-evolved, the flowers providing food for the insects in the form of nectar, and the insects providing transportation for the pollen to another flower's female reproductive parts. This, my friends, is what we call mutualism, the interaction of two organisms which mutually benefits both. Angiosperms reproduce by making flowers that contain the gametophytes. In this case, the sporophyte is made up of the stem and the roots and the leaves and even the flowers, all of the other parts of the plant except the pollen and the ovum, which are the actual gametophytes. Some flowers contain both male and female gametophytes. These are called perfect flowers, no pressure other flowers. Other flowers have both male and female sex organs on the same plant, but in different flowers, and some have male and female flowers on entirely different plants. There are no rules with angiosperms, they're just winging it. To see how flowers work, let's take a look at a perfect flower as an example because a lot of the garden flowers you see have both male and female reproductive parts. Starting from the bottom up, a flower has sepals, which look like leaves or petals, but they're usually green tissue that covered the flower when it was a little bud. The petals are usually colored to attract a certain kind of pollinator, like a flag. The male parts of flowers consists of an anther, which produces the pollen and sits on the end of a long filament attached to the base of the flower. This whole male reproductive setup like this is called the stamen. Now, when it comes to lady parts, in contrast to gymnosperms, angiosperms don't leave their eggs hanging out all exposed. They lock their ovules down in an ovary at the bottom of a vase-like structure, which also has a neck called a style and an opening at the top called a stigma. Now all that's left is to get the male gametes packaged up in their gametophyte, the pollen, and have them carried to the female gametophyte, the ovule, to fertilize them. This is pollination, and flowers do it by luring animals with smells, colors, and food, and in return the animals mix and match the pollen with different individual flowers. Bees are the most famously successful at this, but lots of other insects do it too, as well as birds like hummingbirds and even some bats. So no matter who does it, after fertilization happens, the ovule starts to swell and the ovule wall starts to toughen up because it's going to become a seed. The ovary, meanwhile, starts to grow around it and become the fruit. Now there are a bunch of different types of fruit. Fruit is defined as anything that the ovary, the protection around the seed, turns into, so anything that contains a seed is a fruit. And that's a lot of different things, including many things that we think of as not a fruit. To test your fruit skills, how about a round of fruit and not a fruit? So, which one of these is the fruit and which one of them is the not a fruit? 1, a sandspur you get while rocking around on the beach, or a carrot? Answer: a sandspur, the little annoying thing that attaches to your pants, is actually the swollen up ovary of a flower. A carrot is the root of a plant. A stalk of celery or a piece of dandelion fluff? Fluff. That little piece of fluff is attached to a dry little fruit that contains the seed. Celery is the actual stalk of a celery plant. A strawberry! Or a zucchini. The zucchini! A strawberry is actually the swollen end of the stem of the strawberry flower, so it doesn't contain the seed, it actually has the seeds on the outside. Each one of the hard little things on the outside of the strawberry, those are the fruit. Some people argue about this because what seems more fruity than a strawberry, but zucchinis? Well, they're definitely fruits because they contain seeds. Fruits are important to angiosperms because they like to get their seeds as far away from themselves as possible so that they're not competing with their own offspring. So some fruits can be carried away by the wind while others move around by being totally delicious so that they can be eaten by an elephant and pooped out in an elephant turd far, far away. So that's the steamy sex lives of vascular plants. Ah, that is good. Thank you for watching this episode of Crash Course Biology. It really is like a perfect nectarine. Thanks to everyone who helped put this episode together, including this nectarine. And if you want to go and check out any of the angiospermy mess that is the sex lives of vascular plants, there's a table of contents over there. If you have questions for us, we're on Facebook, we're on Twitter, and we're always in the comments below. And we'll see you next time.